Okay, so I want to go over this paper in Medical Express called "The Brain Benefits of Exercise Can Be Gained with a Single Protein." So it'd be nice in the future if we can just have a pill that we can take that ends up keeping ourselves youthful, especially our brains youthful, because if you get brain damage, very frequently it is a progressive problem, and it ends up getting worse and worse as you age. But exercise is known to be able to improve your cognitive abilities later in life. And so this group has done the study saying what proteins are associated with exercise that are also improving our cognitive ability later in life. And so that is what this press release is going over. So if you want, I'm going to go a little bit. If you want to finish right now, just knowing that, you can leave right now. But I'm going to go through this release, and then I'll go a little bit into the science behind that. If we want to continue, or if you want to continue with me. So here we go. A little studied liver protein, so a protein that is produced within the liver cells or the hepatocytes. This may be responsible for the well-known benefits of exercise on the aging brain, according to a new study in mice by scientists at the UC San Fran Eli and Edith Broad Center for Regenerative Medicine and Stem Cell Research. So, the findings could lead to new therapies to confer neuroprotective effects of physical activity on people who are unable to exercise due to physical limitations. Now, granted, they're saying that this is for people who cannot exercise, but we know for a fact that if we do end up going down this route by developing a product that ends up improving neurodegenerative diseases without exercise, then it's going to be marketed and used for everyone, not just people who can't exercise. And we would also presume that people are going to try to combine it with exercise in order to get more benefit. But I digress. So exercise is one of the best studied and most powerful ways to protect the brain from age-related cognitive decline, and has been shown to improve cognition in individuals at risk of neurodegenerative diseases such as Alzheimer's disease and the frontotemporal dementia. Even those with rare gene variants that inevitably lead to dementia. So there are a few people out here who have genetic disorders that will predispose them and guarantee that later in life they are going to get dementia. And this is well recognized. But even those people, even the people with the genetic predisposition to get dementia, they end up also receiving the anti-dementia health benefits of exercise. Continuing, but many older adults are not able to exercise regularly due to physical limitations or disabilities, and researchers have long searched for therapies that could confer some of the same neurological benefits in people with low physical activity levels. So this is unfortunately the case for many people in the world, that as they get older, they have damaged some part of their body, and that damage leads to a physical disability which prevents them from being able to exercise. And it ends up becoming a positive feedback loop in their life that ends up hurting their body, therefore they cannot work their body, and by not working their body, their brain ends up suffering, and then it could end up becoming a very vicious cycle that they would prefer to get out. But the wonderful thing about a positive feedback loop is if we are able to regulate it and heal it, then the benefit isn't just one order. There will also be multiple orders of effects or positive effects of influencing this cycle. So if we can get people out of that rut, then hopefully there might be long-lasting changes or benefits. Anyways, the new study published July 9th in Science showed that after mice exercise, their livers secrete a protein called GPLD1 into the blood, and levels of this protein in the blood correspond to improved cognitive function in aged mice. So, and a collaboration with the UCSF Memory and Aging Center found that the enzyme, which is this GPLD, is also elevated in the blood of elderly humans who exercise regularly. So this is good. So, but the researchers showed that simply increasing the amount of GPLD1 produced by the mouse liver could confer many of the same brain benefits as regular exercise. So here they've done an experiment where they've upregulated or increased the amount of GPLD1 that is secreted, and this increased secretion has been shown to, or at least in this experiment, showed to increase the cognitive function of the old mice. So let's take a break and look at what GPLD1 is. So here, GPLD1 is also known as phosphatidyl inositol glycan specific phospholipase D, is an enzyme that in humans is encoded by GPLD1 gene, or the GPLD1 gene. Many proteins are tethered to the extracellular face of eukaryotic plasma membranes by a glycophosphatidyl inositol, also known as a GPI anchor. So let's take a step back. A lot of words there. So the cell has a membrane, right? So every cell has a membrane that surrounds it. And these membranes are phospholipid bilayers, and that they have their, on the outsides of the bilayer, you have your phosphate groups, or you have, how do I phrase this? You basically have polar groups, water-loving, hydrophilic groups, and on the inside of those phospholipid bilayers, you have lipids, or fat-loving tails, if you will. And so that means the cell membrane has a fatty inside that protects, or I should say separates, the solution on the inside of the cell and the solution on the outside of the cell. So phosphatidyl inositol, this is like a soap almost. So phosphatidyl, actually, let me look this up so that we have, here we go. So this would be an example of a phosphatidyl inositol, where you have these fatty groups over here, plus a phosphate, and this is glycerol right here, and this glycerol has this fatty acid and this fatty acid associated with it, and then here there will be a phosphate group on the other end of this glycerol, and then this phosphate will also be bound to this inositol. I don't want to call it a sugar, but polyol. So it almost looks like a sugar, but it's not quite. Regardless, this is an anchor in that this group right here is going to insert itself into the fatty portion of the cell membrane. And this phosphate group is going to be sticking out with the other phosphates of the phospholipid bilayer of the cell membrane. And then this inositol group, it has all these alcohol groups on it, which can be covalently bound to other groups or other proteins or other sugars. So this is called an anchor because it can anchor whatever is attached to this inositol. It can anchor it to the cell membrane. So many proteins are tethered to the extracellular or the outside face of the eukaryotic cell membranes through glycosyl phosphatidyl inositol. So this isn't just phosphatidyl inositol. There would also be some sugars that are attached to one or more of these alcohol groups, and you can form a small polysaccharide this way. Regardless, this is where the name GPI comes from. It is glycosylated phosphatidyl inositol, and this serves as an anchor. So this GPI anchor is a glycolipid which is found in many blood cells, and the protein encoded by this gene, GPLD1, this is a degrading enzyme, or it is a GPI degrading enzyme, so it destroys these. So, and remember, this is a phospholipase, so it's going to attack the phosphate group and break it with water. anyways, the protein and so glycophosphatidyl inositol specific phospholipase D1, this hydrolyzes the inositol phosphate linkage in proteins anchored by phosphatidyl inositol glycans, thereby releasing the attached protein from the plasma membrane. So going back here, this GPLD1 is going to come here and break this inositol from the phosphate and the rest of the lipid. So this thereby releases the attached protein from the plasma membrane. So you could imagine if you have a lot of these GPLD1s floating around in the plasma, they're going to go around and be removing proteins from a lot of the surfaces of the cells in your body. So, and apparently this GPLD1 is associated with the improved cognitive function of aging rats. So, and also in humans. In humans, this is expressed in higher levels if you are exercising while you are old. Let's continue. But the researchers showed that simply increasing the amount of GPLD1 could confer those benefits even without exercise. And this sounds great, right? So, what I'm thinking about this is, can I get this to be higher expressed by my diet as well? Does it need to be exercise, or are there different chemicals that I can consume that can help increase GPLD1? Another question I have is, if I can increase GPLD1, will that work holistically, or will that work synergistically with exercise? So can I exercise and take a chemical that can increase this, and will I receive an even greater benefit, or are there diminishing returns or a negative effect by doing that? So anyways, if there was a drug that produced the same brain benefits as exercise, everyone would be taking it. Now, our study suggests that at least some of these benefits might one day be available in pill form, said study senior author Saul Villeda, PhD at this location. So Villeda's lab had previously shown that biological factors present in the blood of young mice can rejuvenate the aging mouse brain. So we might have, if you remember, a few years back, there was some noise being raised over how the blood of young people was very able to, or I shouldn't say young people, but the blood of young mice was able, if it was injected into older mice, it ended up giving or causing youthfulness in those older mice. And so here we can have an example, what I would believe would be a good biophysical explanation as to why those the blood of the young mice was able to confer some youthfulness to the old mice. It might be through this particular protein, which is an enzyme that can theoretically cause youthfulness, right? Anyways, conversely, factors in the blood of older mice can bring on premature age-related cognitive decline in young mice. So I don't really like exactly where this is going, because it might be in the future that young people donate their blood and sell it to older people, or old people will buy young people's blood in order to get some of the health benefits of that. And I mean, that's just something that I could see happening in the future. Regardless, these previous results led Villeda's lab graduate student Elena Horowitz and postdoctoral researcher Chualai, I apologize for butchering this name, but Chualai Fan, who's also a PhD, to pursue blood-borne factors that may also confer the benefits of exercise, which is also known to rejuvenate the aging brain in a similar fashion to what was seen in the lab's young blood experiment. Anyways, Horowitz and Fan took blood from aged mice who had exercised regularly for 7 weeks and administered it to sedentary aged mice. And they found 4 weeks of this treatment produced dramatic improvements in learning and memory in the older mice, similar to what was seen in the mice who had exercised regularly. When they examined the animals' brains, they found evidence of enhanced production of new neurons. That's excellent. That's a good way to demonstrate that the nervous tissue is actually improving and creating new growth. In the regions known as the hippocampus, as well as a well-documented proxy for the rejuvenating benefits of exercise. So, the hippocampus, one of the sections of the brain that is in the middle of it, to see benefits in the hippocampus is usually, or would be indicative of the entire brain really functioning or healing in some fashion. Continuing, and I believe the hippocampus is important with secretion of some hormones, right? Let me look real quick just to see. Plays important roles in the consolidation of information, short-term and long-term, into long-term memory and in spatial memory. So this is one of the first regions in the brain to be damaged in people with Alzheimer's and other forms of dementia. So it seems to be close to the hippocampus is here, and the hippocampus. Okay, and here's the thalamus. Okay, so yeah, we'll leave that at that. I'm not a neurosurgeon, so we will continue reading. Anyways, just to discover what specific biological factors in the blood might be behind these effects, Horowitz, Fahn, and colleagues measured the amount of different soluble proteins in the blood of active versus sedentary mice. And so they identified 30 candidate proteins. So this is one of the things that biochemists frequently do, is we will have the subjects live their different lifestyles, or we will mutate one subject while leaving the others controlled. We'll just have a control and have something else, and then measure the differences of the proteins that are expressed or secreted, or the chemicals that exist in one versus the other. And if there are significant differences, this can tell you which ones are important in that difference. Anyways, these people identified 30 candidate proteins, and 19 of which, to their surprise, were predominantly derived from the liver, and many of which had previously been linked to functions in controlling the body's metabolism. Well, the body's metabolism thing makes sense to me. So if you are exercising a lot, you're probably going to have to express proteins that are associated with proper metabolism, because you probably have a higher metabolic rate. And so those are, I would presume, would be important proteins for that to function properly. Now, however, 19 of which, to their surprise, was predominantly derived from the liver. Now, this is somewhat interesting. It seems as though the liver is playing a very large role in metabolism, which we knew that already. We know that the liver plays an important and large role in metabolism. However, to see that 19 out of 30 are coming from the liver, this is a little, that is surprising, right? Anyways, 2 of these proteins, 2 out of the 19, GPLD1 and PON1, stood out as particularly important for metabolic processes, as the researchers chose to study, GPLD1 in more detail because few previous studies had investigated its function. So it could also be that PON1 also is very important in this process, which let's search it up just to see what exactly it is. So this is a serum paraoxanase and aryl esterase, also known as, this is what it looks like. This is the structure of its function. And this is an esterase, which means it breaks up ester groups, and more specifically, paraoxanase activity. And these are, let me see if they are involved in the hydrolysis of organophosphates. So let's see if there's an example of what that, no, they don't have an example. So how do I phrase that? Basically, this is a catalytic enzyme that can break up a chemical bond, would be the best way to describe it, which is very similar to the GPLD1. Anyways, these stood out as particularly important for metabolic processes, and so this this group decided to study GPLD1 into more detail because it was a unique, or I should say a less studied protein. Anyways, we figured that if the protein had already been investigated thoroughly, someone would have stumbled upon this effect. So I like to say, if you're going to take a risk by exploring something new, you might as well go big. So yeah, that is a good presumption to make. Granted, if no one was looking for this particular activity upon one, then they might have missed it as well. So I would highly recommend to this lab, or if you're looking for different research projects and this is in your field, study PON1 as well. See if that is also conferring similar benefits. Anyways, this team found that GPLD1 increases in the blood circulation of mice following exercise, and GPLD1 levels correlate closely with improvements in the animal's cognitive performance. So we notice this in humans as well, and that when you do exercise, you end up learning better following that exercise. So is it possible that GPLD1 in humans is associated with this improved cognitive function, or also this PON1? Is this also associated with improved cognitive function? I mean, it is correlated just because it is expressed higher after exercise. Maybe. Anyways, let's continue. See if we learn any more. So, to test whether GPLD1 itself could drive the observed benefits of exercise, the researchers used genetic engineering to coax the livers of H mice to overproduce GPLD1. So there are ways that you can mutate or genetically engineer the DNA to express more of a particular gene. And so they did this with the liver cells of these mice. They then measured the animal's performance in multiple tests that measure various aspects of cognition and memory. And to their amazement, 3 weeks of the treatment produced effects similar to 6 weeks of regular exercise, paired with dramatic increases in new neuron growth in the hippocampus. Now, let me see. So I would have to look at to what this genetic engineering was, because it sounds like this is something that was done after the mice have already been conceived and born. So yeah, I am going to end up looking at this paper later, which this... Oh, no, not that. Was it... Okay, well, I had the paper up earlier, which... Oh, yeah, it's right here. So, more information, blood factors transfer benefits of exercise on neurogenesis and cognition in H brain. So this is the paper. So if you want to read it yourself, you can go here. And actually, yeah, it's also, I believe, the full text. Oh, no, you would need to have a subscription, but I'm going to ask. Oh, that's okay. Regardless, we'll keep on going. But if you want, you can go to this website and read it for yourself. Anyways, to be honest, I didn't expect to succeed in finding a single molecule that could account for so much of the benefits of exercise on the brain. It seemed more likely that exercise would exert many small, subtle effects that add up to a large benefit, but which would be hard to isolate, Villeda said. When I saw these data, I was completely floored. So this is something that we very... it's what he's saying right here is not crazy, or it's not, I should say what he's saying makes sense. What he's saying right here was very intuitive, that we would hope, since exercise does so much to the body, we would hope, or we would presume, that exercise is changing a lot of different metabolic pathways in the body, and this is creating the differential expression of a lot of different proteins and chemicals in the body. And any one of those things would be very difficult to isolate. However, in the long-term benefit, all of these small things would theoretically come together and make a very large effect, which in this particular case, we'd say these are all creating improved cognitive function. But it appears as though that's not the case. It appears as though there is only a select few proteins that are doing the majority of the benefit. So here they're saying the majority of the benefit might be just from one singular protein, which would be phenomenal, because that means that the less complicated a system is, the easier it is to manipulate it for your own benefit, or develop pharmaceuticals, or develop just treatment protocols. Hell, even in regards to developing new tests and developing new strategies, we could use this as a biomarker. We could take blood samples from people, quantify how much of this GPLD1 is in their blood, and then we can correlate that with different lifestyles, and correlate that with different chemicals or different foods that they eat. And then we could use this as a way to correlate lifestyles with, or different variables with GPLD1 expression. Now, and this could be beneficial for longevity, right? If we can just learn the best ways of existing and living in this world that can increase our GPLD1 levels, well, we might want to optimize our lifestyles in accordance with this GPLD1 concentrations. Now, granted, that is taking it to the extreme, right? We don't want to optimize for just one protein. We don't want to optimize for just one variable in our lives. However, this could be one of the variables that we want to try to optimize in concert with other variables. So this is excellent. Really, this truly is going to be, it's already been published, but if this is verified through multiple other studies, this might very well change how humans live into adulthood. Anyways, through these proteins, the liver is responding to physical activity and telling the old brain to get young. So this is a remarkable example of a liver-to-brain communication that, to the best of our knowledge, no one knew existed. So it makes me wonder what else we have been missing in neuroscience by largely ignoring the dramatic effects of other organs might have on the brain, and vice versa. So yeah, this is something that, this is one of the downfalls, or one of the problems with being way too specific in your field of study, because if you try to isolate your organ of interest or your protein of interest to only what is known, or what connections are directly made to that, your field of study can become a little stifled, because you're trying to learn more and more about these connections that we already know of. But if there's a completely different connection somewhere, you really just have to stumble upon it, which is apparently what is happening here as well, where he just had to, or I don't know if it's a man or woman, I apologize if I'm misgendering, but we don't know. If you don't take that first step, or if you don't bring in something completely out of the box into your field, you might become stifled. And so, yeah, this is remarkable that this happened. Anyways, further laboratory experiments have shown that GPLD1 produced by the liver does not pass through the so-called blood-brain barrier, which protects the brain from the toxic or infectious agents in the blood. Instead, the protein appears to exert its effects on the brain through pathways that reduce inflammation and blood coagulation throughout the body. Anyways, both blood coagulation and inflammation are known to be elevated with age, and have been linked to dementia and age-related cognitive decline. So apparently, this protein cannot pass the blood-brain barrier, but it is able to exert its effects on other tissues, right? And apparently the effects on the other tissues are able to benefit the brain as a whole. Now, granted, we don't know precisely. The blood-brain barrier is not perfect. It really is not a perfect barrier. So there is the possibility that some of this GPLD1 is leaking into the cerebrospinal fluid, right? But yeah, hopefully, or maybe in its current form, the form that they're studying, maybe that doesn't get into the cerebrospinal fluid easily, but perhaps some further processing then allows it to be transferred easily. We don't know. They haven't looked that up yet. What an experiment I would, personally do is um, see if there is any GPLD1 in the brain tissue, right? So you can take the mice, you can euthanize them. They've lived long, healthy lives, right, because of the high expression of GPLD1. But you can do a immunohistochemistry, or you could take a microscope slide of the brain tissue of these mice and see if there are significant differences in GPLD1 within the brain tissue. Now, granted, um, you might not see any in the brain tissue, right? So that might be something that you also have to consider. Okay. Anyways, the lab is now working to better understand precisely how GPLD1 interacts with other biochemical signaling systems to produce its brain-boosting effects in hopes of identifying specific targets for therapeutics that could one day confer many of the protective benefits of exercise for the aging brain. Awesome. So, with that, I think I'm going to head on out, but yeah, I'm very excited to see where this ends up going. Now, my presumption, well, for one, these where is it at? These phosphatidylinositol groups, so the glycosyl phosphatidylinositol, these do also play a role in a lot of neurons, right? So a lot of neurons will also have these phosphatidylinositol groups on them. So is it possible that maybe there is some leakage of this GPLD1 into the brain, which allows it to act upon the neurons as well? Yeah, we'll have to see. Perhaps for people who are older, perhaps their blood-brain barrier isn't as effective, and so this is able to leak into it for older people, whereas younger people might not have that effect as much. Who knows? This is a completely new field, and so I'm really, really happy that these people were able to stumble upon this. So anyways, with that, I'm going to head on out, but thanks for watching, and hopefully I will see you tomorrow.